All right, so the order of draw. This is important. It's important that we know and we learn and we memorize the order of how we draw blood based on the test and the tubes that we have to use. Um, there's a specific standard, and what this does is help from contamination of additives into other tubes and any errors in the results of the draw. So we're going to get into what that order may look like. CLSI has developed a set of standards dictating the proper order of draw for a multi-tube draw. So this means multiple tests or more than one test is ordered, and it requires more than one tube, one different kinds of tubes, okay? So the order is the same for syringe samples as for direct filling from a multi-sample needle, okay? So it doesn't matter, it's going to be the same either way. The order of draw standards have undergone several revisions within the last decade, and not all institutions have adopted the most recent set of standards, but these standards are what we are going to use. So I'm going to go through those with you guys now. This is really important. We'll help you with this. This is something you need to know correctly how to draw when you have multiple tests. So this is the correct order of draw based on the CLSI standards, okay? So we're going to draw the sterile tube first. That's the yellow tube. Then the coagulation tube, light blue. Then the serum tube, red. Then the SST, which is the red tiger top or gold top. The PST, light green top or lithium heparin. The green, which is the dark green top or sodium heparin. The EDTA, which is the lavender. And then the gray, okay? So that's the order in which we're going to. So it is important that when we're collecting multiple tubes of blood that we draw in the collect order. So we're basically doing blood cultures. Light blue top, red or speckled tubes, green tubes, lavender tubes, or and then gray tubes. So the reason why we do blood culture tubes first, they are sterile. They're sterile, all right? So this is why we want to do them first. What happens is the reason we do them first is it prevents the transfer of unsterilized material from one tube into our sterile tube. So we're going to do that sterile tube first, all right, so the blood culture tubes. From there, we're moving to the light blue topped tubes. These are coagulation tests. These tubes are always drawn before tubes containing other kinds of anticoagulants or clot activators, because these additives can contaminate this tube, right? So these, the coagulation tests, we don't want an anticoagulation additive to sneak in this one, because that can result in an error in the results. If the coagulation tests are the only test ordered, you only need a light blue topped tube. Your institution, so depending on where you work, may have you draw a plain red top tube first and discard it, all right? And then draw your light blue top, all right? So that is going to depend on your institution. The discard tube may be a plain red glass tube or a specifically manufactured plastic tube without any additive. So what they're having you do, is draw from a glass red tube. Remember, that has no additives in it. Discard that and then draw from the light blue. And once again, that's based on your institution. So that's something you need to pay attention when you go get a job here as a phlebotomist. If you're drawing with a butterfly, you must draw a discard tube to draw out the air in the tubing and prevent a short draw. That's really important, okay? So you're drawing an extra tube that you're going to discard. So that's what we call a discard tube. After you've done our blood culture tube, our light blue tube, now we're going to move to that glass red top tube, all right? Or plastic red top tubes may be drawn now. Then we're going to go to the red gray or the gold, all right? These tubes are plastic gold top tubes. These ones are going to be next. These contain clot activators, which would have interfered with the other tubes if we would have used it, specifically the light blue. After the red, gray, or the gold, you're going to move on to the green tubes. The heparin from the green tube is basically less likely to interfere with the EDT containing tubes than vice versa. As we move on, we're going to move on to the lavender top tubes. The EDTA binds many metals, so things like calcium, and what that can do is it can cause problems with a lot of test results, all right? So this is why we draw it near the end. Lastly, we have the gray top tube. All right, this tube contains potassium oxalate. All right, this would elevate the potassium levels measured in any electrolyte analysis. And oxalate can damage cell membranes. So this is why we draw this last. All right, so this is the order of the draw and the reasoning why we draw in this order. This is something that's going to be really important for you to know. We will also give you guys some study materials, some little handy like handouts that will help with this order of the draw and for you to understand it.